Hello, thanks for visiting my poster titled Spatial Calibration for 3D Freehand Ultrasound via Independent General Motions. My name is Qianqian Cai, and I am a PhD student at North Carolina State University in Raleigh, North Carolina, United States. This work was in collaboration with the University of Toledo and UNC Chapel Hill. 3D freehand ultrasound reconstructs the 3D structure of the imaging object by estimating the trajectory of ultrasound images via a tracked ultrasound probe. Figure 1 shows our previous result to reconstruct a printed 3D fetal phantom based on a linear scan. There are multiple advantages of 3D freehand ultrasound. First, it is cost-effective. Second, the image quality is less dependent on the sonographer's skills. Third, it is more intuitive to understand the imaging object based on a reconstructed structure compared with a series of 2D ultrasound images. However, there are still unsolved issues behind this concept, which includes the spatial calibration that helps to transform the marker's trajectory to that of the ultrasound images, the temporal calibration that synchronizes the tracking system with the ultrasound system, and the low imaging resolution along the elevational directions. Therefore, the objective of this work is to first find out the fixed image-to-marker relationship and the scaling ratios along the axial and lateral directions of the ultrasound images, and then to reconstruct a 3D phantom based on the tracking data. In this study, we adopted the camera tracking. Camera tracking requires markers attached to the tracking object. Since the ultrasound imaging plane is not physically existed, markers are usually attached to the probe. But due to the angle effect errors, trajectories of the ultrasound images can be different from that of the marker sets during the freehand motion of the probe. Therefore, calibration is required to estimate the trajectory of the ultrasound images in relation to the camera based on the recorded motion of the marker set. Figure 2 and Figure 3 shows the transformations and coordinates involved in a general calibration process. Let's look at Figure 3. There are 3 coordinates here. One is image coordinate I, one is marker coordinate M, the other is camera coordinate C. And there are also 2 transformations. One needs to transform the image coordinate I to marker coordinate M using the matrix T I to M, which is unknown, and then to transform the marker coordinate M to camera coordinate C using matrix T M to C. And these matrices can be calculated from the camera reading. Current calibration method requires imaging test using phantoms like wild phantom, point phantom, or plane phantoms. Take a wild phantom as an example. There is usually an automatic segmentation or a semi-automatic segmentation of intersects between the ultrasound imaging plane and the wild phantom. However, recognition of isolated points in ultrasound images is not very reliable due to the image resolution. Therefore, we proposed our method to solve the spatial calibration issue via independent general motions. A probe motion consists of both translation and orientation is a general motions. 2 general motions that do not share parallel axes are independent. It is proved that with at least 2 independent general motions, the spatial calibration problem can be converted into a math problem, which is to uniquely solve a matrix equation in the form of AX equals XB. Next, I will explain how to derive this equation. In Figure 4a, if we look at the loop that transforms the coordinate I2 to M1, we can find 2 different passes. One is from I2 to M2, then to M1. The other is from I2 to I1, then to M1. The 2 different passes form the equation in the form of AX equals XB, where X represents the relative relationship between the image and the marker set. In the loop, Ti1 to M1 and Ti2 to M2 is equal, both of them represent the fixed unknown relationship. And the A matrix in the equation represents the relative motion between the two marker sets M1 and M2, and it can be calculated from the camera. The difficulty here is how to accurately estimate or measure the relative motion between I1 and I2, which is a transformation matrix TI2 to I1. In order to determine the relative position between the two ultrasound images, we need to determine the position of the central imaging plane of the ultrasound beam. In our experiment, we used 3 points to determine the plane position. The first 2 points are the 2 middle points of the probe's short edges as they are shown in figure 4b. In order to determine the position of the third point, we designed an imaging test. In the imaging test, we adopted a bead phantom with a diameter of 7.95. We then placed the bead phantom under the butterfly IQ probe and translate it along the elevational axis until we obtained a high-quality ultrasound image that reflected the real size of the beam, as it is shown in figure 5b. As we know the position of the 3 points, we placed markers there, and those markers form a marker set to be tracked by the camera. Then we can estimate the relevant motion between the ultrasound images based on the relative motion between the 2 marker sets. And then we obtained our B matrix. With A and B known, we were able to solve the equation to obtain the fixed relationship between images and the marker set. So our conclusions here are: the fixed image to marker relationship can be estimated in a closed form manner by solving the equation given at least 2 independent general probe motions. And calibration errors caused by the ultrasound beam thickness can be minimized as the small size bead phantom placed close to the central imaging plane of the ultrasound beam along the elevational axis. This work was supported by Bill and Melinda Gates Foundation. The authors want to thank Butterfly Network Company for their support. Thank you again for visiting my poster. Please visit our lab website if you are interested in our publications.